Hi everyone, thank you for joining the molecular devices cellular imaging webinar series. Will be starting in just 1 minute. I hope everybody can see that slide. I will be going through a short introduction, and you'll be introduced about how to ask us questions and comments. So today we are going to be talking about identification of selective inhibitors of the STAT3 signaling pathway. I'm Mr. Chandy from Molecular Devices. I am your host and the product manager for cellular imaging at Molecular Devices. So for those who actually can't hear me, this is you should have been seeing the slide earlier, but it's for those who can't hear me, this is just a visual to help you get to the audio. There's two audios. You can either be teleconferencing in with your phone or selecting an integrated VoIP. And if anybody has any technical issues with slides or audio, there's a help number from WebEx, who's the host. So, as I mentioned, here's a picture of me. I'm the product manager for the Image Express and Med Express line of products here at Molecular Devices. We have Paul Johnson with us, who is going to be discussing his STAT3 screen that he's implemented with the Image Express Ultra. Paul Johnson is a research associate professor in the Department of Pharmaceutical Sciences, School of Pharmacy at the University of Pittsburgh, as a member of the Drug Discovery Institute in the School of Medicine. Dr. Johnson has 21 years of drug discovery experience in the pharmaceutical, biotechnology, and academic sectors. His career spans a number of small and large pharma companies, and then in 2005, he joined the University of Pittsburgh to help design and build infrastructure for high-throughput drug discovery screening center. And it's quite an elaborate center with compound inventory, lens management, compound storage, and of course doing a number of screens. Since joining Pittsburgh, he has led 15 screening campaigns and continues his research in using HTF and HCF assays. Anytime during the webinar, please submit Q&A, and we have Jane Hesse, our application scientist, will be compiling the questions and asking either Paul or myself questions at the end. Go to the Q&A button to expand the Q&A window. Type in a question. Choose all the panelists so that all of us will see that question, and hit the send button. This might look a little bit different from what you're seeing right now. There's some updates to WebEx, but generally it's the same workflow. So I wanted to tell you a little bit about, for those on the line who aren't familiar with high-content screening, high-content screening essentially uses a sophisticated automated microscope to be able to, that enables hundreds of applications in the screening mode. This slide shows you some of those, just a sampling of those applications, anywhere from receptor endocytosis, angiogenesis assay, sort of gross morphology with spheroid analysis, neurite output. There's a lot of people doing neurite output screens for neurodegenerative diseases, and micronuclear formation, where you're looking for these small little buds that show up on the nucleus after they've divided. The high-content screening workflow is typically you acquire images. This is after you drug the assay. You need some tools to convert those images that you acquire to numbers, and that's image analysis. There's a data analysis step where you need to make sense across a screen of the numbers you get. And there needs to be some type of storage and storage management as a back end to manage the data that you acquire, your analysis, and your numerical values. With the Method Devices imaging solution, we have a wide field product imaging first microsystem, and at the focal, the one that Paul's going to be talking about, the imaging first ultra system. These are all connected, can be connected together with a data management solution we call MC Store, and that allows our MetaExpress and MetaExpress PowerPoint software, which converts those images into numbers, to be unified with QtExpress for data analytics. So I want to give you a little background on the ImageFirst Ultra, since that's the product that Paul has used for this particular screen. It is an automated high-speed confocal point scanner. It's not sitting there for no line scanning. It's true point scanning. With an optimized assay, typical assay in-house, we can probably collect 5 3D4 plates an hour. That's one site per well. The benchtop system that can have about up to 4 lasers to be able to go from DAPI to Sci-5. At any one time, you can have 4 objectives in the instrument, going from 4x to 100x. And of course, there's a wider variety of objectives available from us. The ImageFirst Ultra confocal system allows for configure detection pinhole, so allows fine-tuning your resolution for the thinner sections. You can configure your scan length, since it's a scanning confocal, where to optimize either for speed or cell count or imaging long structures like neurons. You can, depending on the objectives you have, either simultaneous or sequential imaging with up to 4 PMTs. There's robotic tape handling. You'll see that Paul has an automated workflow, and it's custom engineered for optimal performance, including laser autofocus, designed for high image quality, precision linear motor stages, and it's a reliable product. So here is an example of a very long scan with GFP, able data arrestin, and DRAC5 to show that you can scan up very long, up to 1 to 32 aspect ratio long images. And again, you do that to optimize for speed or long objects. So that's just a short introduction, and now I'd like to pass the presenter ball over to Paul to talk about his STAT3 screen. And
I'm going to be talking about 97,000 compound high content screening campaign, where our goal was to identify selective inhibitors of STAT3 signaling pathway in head and neck cell, squamous cell carcinoma cell line. So the signal transducers and activators of transcription there are 7 members of the STAT family. STAT1, 2, 3, 4, 5A, 5B, and 6. They are cytoplasmic transcription factors regulating cytokine and growth factor gene expression. And they have a common domain structure. They've got an amino-terminus STAT dimerization domain, a coil-coil protein interaction domain, a central DNA binding domain, Stark homology domain. They have conserved tyrosine residues. Phosphorylation of these tyrosines triggers dimerization, translocation of the nucleus, and activation of transcription. They also have phosphatidylserine residues that also get phosphorylated that modulate transcriptional activity. And then there's a carboxyl-terminus transcription activation domain. STATs 1, 3, and 5 are each activated by a large number of cytokines, whereas STATs 2, 4, and 6 are activated by relatively few. My slides don't seem to be advancing.
Well, I have to go to the bottom left, the little very faint left and right arrow down the bottom. There you go.
I see it. Thank you. Okay, so the reason we're interested in STAT3 is there's a large body of evidence implicating STAT3 as a proto-oncogene. It's essential for VSARC-mediated transformation, and if you make a constitutively active form of STAT3, it mediates cellular transformation. STAT3 upregulates apoptosis inhibitor genes, cell cycle regulators, inducers of angiogenesis and expression of matrix metalloproteinases. And activated STAT3, although there is no currently known, there are no mutations that constitutively activate STAT3. Activated STAT3 is present in many cancers, and it directs tumor cell proliferation and survival, angiogenesis, alters the tumor microenvironment, promotes metastasis, and also leads to suppression of anti-tumor immune responses. This is just a table taken from a review that Jenny Grandis and I wrote on the role of the STAT3 signaling pathway as a therapeutic target in cancer, and you can see that there are a whole list of cancers that are characterized by elevated STAT3 expression or activity, and activated STAT3 is implicated in poor prognosis, and there are a number of abnormalities. As I said, STAT3 itself is not mutated, but there are a number of abnormalities in the STAT3 signaling pathway that lead to elevated STAT3 activation, including elevated EGF receptor expression, constitutively activated EGF receptor, overexpression of the SARC family kinases, hyperactivated Janus kinases, and elevated TGF-alpha or IL-6 levels. And activated STAT3 has been implicated in a number of xenograft models. Now, we're going to focus primarily on head and neck cell squamous cell carcinoma. There's a little bit of background that's taken from the ACR website, 2011, on head and neck cell. It's the sixth most common non-skin cancer in the world. There are approximately 600,000 cases per year with a 50% mortality rate. The incidence is implicated with alcohol use and smoking, and there's also a high incidence or a high increase in head and neck cell cancers attributable to human infection with human papillomavirus. And it's a big problem in patients because recurrence, metastasis, and second primary cancers account for the majority of the deaths from this invasive cancer. There's a lot of diversity in the genetic nature, and it represents a major challenge for effective therapies. The current frontline therapies are surgical resection and chemoradiotherapy, surgical ablation and radiation and cytotoxic drugs are clearly non-specific, and the overall cure for head and neck cell cancer is less than 50% of patients. And of course, the current treatments are limited by toxicities and side effects and impaired quality of life and reduced survival. Okay, so this is just a cartoon that is showing the STAT3 signaling pathway, the canonical. I'm going to be talking about 97,000 compound high-content screening campaign, where our goal was to identify selective inhibitors of STAT3 signaling pathway in head and neck cell squamous cell carcinoma cell line. So the signal transducers and activators of transcription have a There are 7 members of the STAT family, STAT1, 2, 3, 4, 5A, 5B, and 6. They are cytoplasmic transcription factors regulating cytokine and growth factor gene expression. And they have a common domain structure. There's an amino-terminus STAT dimerization domain, a coil-coil protein interaction domain, a central DNA binding domain, Stark homology domain. They have conserved tyrosine residues. Phosphorylation of these tyrosines trigger dimerization, translocation of the nucleus, and activation of transcription. They also have serine residues that also get phosphorylated that modulate transcriptional activity. And then there's a carboxy-terminus transcription activation domain. STATs 1, 3, and 5 are each activated by a large number of cytokines, whereas STATs 2, 4, and 6 are activated by relatively few. My slides don't seem to be advancing.
Oh, I have to go to the bottom left, the little very faint left and right arrow down the bottom. There you go.
I see it. Thank you. Okay, so the reason we're interested in STAT3 is there's a large body of evidence implicating STAT3 as a proto-oncogene. It's essential for V-SARC-mediated transformation, and if you make a constitutively active form of STAT3, it mediates cellular transformation. STAT3 upregulates apoptosis inhibitor genes, cell cycle regulators, inducers of angiogenesis and expression of matrix metalloproteinases. And activated STAT3, although there is no currently known, there are no mutations that constitutively activate STAT3. Activated STAT3 is present in many cancers. It directs tumor cells towards proliferation and survival, angiogenesis, alters the tumor microenvironment, promotes metastasis, and also leads to suppression of anti-tumor immune responses. This is just a table taken from a review that Jenny Grandis and I wrote on the role of the STAT3 signaling pathway as a therapeutic target in cancer. And you can see that there are a whole list of cancers that are characterized by elevated STAT3 expression or activity. Activated STAT3 is implicated in poor prognosis, and there are a number of abnormalities. As I said, STAT3 itself is not mutated, but there are a number of abnormalities in the STAT3 signaling pathway that lead to elevated STAT3 activation, including elevated EGF receptor expression, constitutively activated EGF receptor, overexpression of the SARC family kinases, hyperactivated Janus kinases, and elevated TGF-alpha or IL-6 levels. And activated STAT3 has been implicated in a number of xenograft models. Now, we're going to focus primarily on head and neck cell squamous cell carcinoma. There's a little bit of background that's taken from the ACR website, 2011, on head and neck cell. It's the 6th most common non-skin cancer in the world. There are approximately 600,000 cases per year with a 50% mortality rate. The incidence is implicated with alcohol use and smoking. And there's also a high incidence or a high increase in head and neck cell cancers attributable to human infection with human papillomavirus. And it's a big problem in patients because recurrence, metastasis, and second primary cancers account for the majority of the deaths from this invasive cancer. There's a lot of diversity in the genetic nature, and it represents a major challenge for effective therapies. The current frontline therapies are surgical resection and chemoradiotherapy, surgical ablation and radiation and cytotoxic drugs are clearly non-specific. In the overall cure for head and neck cell cancer is less than 50% of patients. And of course, the current treatments are limited by toxicities and side effects and impaired quality of life and reduced survival. Okay, so this is just a cartoon that is showing the STAT3 signaling pathway, the canonical STAT3 signaling pathway, and activation, both activation and endogenous regulation. So there's a whole range of growth factor receptor tyrosine kinases, cytokine receptors, SARC family kinases, and GPCRs that have been implicated in the activation of STAT3. So activation of growth factors and cytokine receptors leads to recruitment of non-phosphorylated STAT3 in the cytoplasm to the activated receptors through their SARC homology domain. They get phosphorylated on specific tyrosine residues that triggers dimerization, translocation, passage through the nuclear pore complex, and DNA binding, ultimately activating gene transcription. And it's an important signaling pathway. There are a number of STAT3 regulators, suppressors of cytokine signaling, the SOX family, the protein inhibitors of activated STAT, and a number of protein tyrosine phosphatases that have been implicated in dephosphorylating STAT3. There have been a number of strategies to try and that people have tried to inhibit STAT3 signaling, including inhibition of phosphorylation or activation of STAT3, and there are a number of compounds out there that have been approved for inhibiting receptor tyrosine kinases, the JAK kinases, and SARC family kinases, and a number of those are in clinical challenges. However, there's modest efficacy, there's often development of resistance, and there are some challenges associated with those treatments, myelosuppression, GI toxicity, and there are also concerns about kinase selectivity and cardiovascular toxicity. Another strategy that people have adopted are to try and inhibit the dimerization through the SARC homology domain, and a number of peptides have been developed that do indeed inhibit those. So there are peptide aptamers and G-quartet oligonucleotides and small molecule peptidomimetics. However, the efficacy of those compounds are limited by poor cell permeability and metabolic stability, and there are issues with selectivity for specific SH2 domains. An emerging area of interest is strategies to inhibit nuclear import or export. I'm not really going to talk too much about this, but that's an emerging area that people are trying to come up with inhibitors for. However, the multi-component nature of the nuclear pore and translocation of the STAT3 through that, there are concerns about specificity or selectivity there. The double-stranded DNA decoys are an effective strategy for inhibiting activated STAT3 binding to DNA. And in fact, Jenny's group have developed a double-stranded DNA decoy, and they've done some clinical trials. Unfortunately, they have, because of the poor cell permeability and metabolic stability of the DNA decoys, they have to do direct intratumor injection, although the group are developing strategies to improve the bioavailability and targeting of the double-stranded decoys. There are a number of natural products, including butysterone, inoculol, curcumin, and you can see them listed here, that have been implicated in inhibiting the STAT3 signaling pathway, but they've also been implicated in inhibiting the whole other range of signaling pathways. So there are concerns over specificity, potency, and efficacy. So the strategy that we came up was to develop a high-content screening assay taking advantage of antibodies that specifically stain phosphotarcine 705 on STAT3. But we wanted, and we're aware of, there's quite a lot of data in the literature implicating STAT3 as an oncogene, but also a very related signal transducer and activator, STAT1, is a tumor suppressor. And, you know, there's a lot of data now that says that STAT1 activation favors cell cycle arrest, apoptosis, and anti-tumor immunity. And so the ideal inhibitor would be one that selectively inhibited STAT3 but did not inhibit STAT1. And although STAT3 inhibitors have been identified by target-based approaches, you know, using fluorescence polarization assays looking for compounds that bind to STAT3, specifically the peptides and peptidomimetics that block STAT3 dimerization, they often exhibit poor cellular activity, lack selectivity, and haven't really progressed into the clinic. So our strategy was then to develop a STAT3 high-content screening, primary screening assay, and incorporate into that testing paradigm a phosphostat1 selectivity assay in the same head and neck cells. And by using those two assays, identify selective STAT3 pathway inhibitors that might have improved therapeutic potential. Okay. So here I'm just listing the components of an automated high-content screening assay development, sample preparation, image acquisition, image analysis, and data analysis. And I've listed just a brief summary of some of the major factors that are involved in each of these steps. And I'm going to take you through the assay development for the STAT3 screening assay and hopefully illustrate some of these issues for you. So one of the first things, and here I'm showing some data generated in Jenny Grandison's group by Malabika Sen. She took some of the head and neck cell tumor cell lines that they had available to them, and she looked at both total STAT3 levels and phosphotarcine 705 levels by Western blotting. And indeed, we ended up picking the Cal-33 cell line as a good cell line to go on to develop the screening assay. One of the issues that we first encountered when we were developing the assay, however, was we had to identify good antibodies that worked in our imaging applications, and we found that there was a distinct difference depending on how well the antibodies worked, depending on which method of cell permeabilization we used. And here I'm showing you 3 different antibodies that we tested. Our standard permeabilization method, which is 0.5% Tritenex 100 versus 90% methanol. And you can clearly see that permeabilization with the methanol provided a much stronger signal. And so what we did then was the usual catalog hopping. We went and I identified which companies had phosphotaricine and total and phosphorylserine STAT3 and STAT1 antibodies and a number of other STAT antibodies. And I would call out and say thanks to some of the folks at Beckton, Dickinson, and Thermo Fisher who provided some of these antibodies to us free while we wanted to test them. So we tested a variety of, we looked at HeLa cells and a couple of other head and neck cell lines, UM22B and 1483s, and tested these different antibodies for reactivity with both the STAT3 and activated STAT3, activated with IL-6. These are our two IXU Image Express Ultra platforms in the lab, and we've got a CRS Catalyst robot hotel that the robot pulls the plates off the hotel, and we can actually run either on one instrument alone or on both instruments. And as Grisha's already told you, it's an automated point scanning confocal imager. We've got 4 laser lines, 4 ejector turrets, 4 PMT detectors, and we have both the MedExpress and Acuity Express software and NBC store database. We also purchased the PowerCore distributed for post-acquisition analysis to speed it up, and we have 10 image analysis modules. And actually, these are the image analysis modules that we have on hand. I'm really going to talk mostly about the translocation enhanced image analysis module and the multi-wavelength cell scoring towards the end. These are 2 image analysis modules that we use for the screen, the assay development and the screening. This is just a summary of the high-content imaging acquisition. We use the 20x.45 NA, extra long working distance objective, the infrared laser autofocus. Obviously, we use the 405 and the 488 lasers, quad filter cube, and we acquired 2 fluorescent channels, acquired sequentially, the Hertz channel. We had a Z offset of minus 6.98 microns from the autofocus point, and we used the 405 laser, 10% power, and a PMT gain of 550. For the phosphotyrosine 705 50 channel, we had a Z offset of 12.9 microns above the autofocus point, or away from the Hertz Z offset, with the 488 laser at 10% power and a PMT gain of 625. We took 2 images or fields of view per well, and it took us 90 minutes to scan a full 384 watt plate using these settings. Here's just some images. I'm showing you the media control and the IL-6 controls. We've got the Hertz GFP channel 1, the phosphotyrosine in channel 2, and then a composite image or a color composite of these 2 images. And you can clearly see that IL-6 gives you a nice induction of phosphotyrosine, primarily located in the nucleus. This is how we configured the translocation enhanced image analysis module. We set up approximate widths and intensities above background with a minimum area, maximum area, and for the nuclear channel, and then in the Finsky channel, we eroded the detected nucleus by 1 pixel, and then we set up a mask in the cytoplasm 1 pixel away from the detected edge of the nucleus, and 3 pixels or 4 pixels in width, so 3 microns in width. So this is showing you some images of the median IL-6 treated cells, just blowing up portions of the nuclear channel and the phosphotyrosine channel, and then the inner masks, which are in the lighter colors, the light red and the light green, or the outer masks, or cytoplasmic mass, which are in the dark green or the dark red. And then to the side, I'm just showing you some of the parameters that the image analysis module gives you. You've got the cell count derived from channel 1, the average phosphotyrosine STAT3 intensity in the cytoplasm, and then the average phosphotyrosine intensity in the nucleus, and then you can also express that as a ratio of the nucleoside intensity ratio. In addition to the average intensity readings that I'm showing here, you could also get the fully integrated intensity readings. We actually ended up using the average phosphotyrosine 705 intensity in the nucleus as the main parameter for the high content screen. Okay, here's some assay development and optimization experiments that we did for both the phosphotyrosine STAT3 and the phosphotyrosine STAT1 assays. We're looking at the ability of different cytokines, mediating interferon gamma, IL-6, and EGF to induce STAT3 activation in the same cytokines for inducing STAT1 activation. You can see that IL-6 is the most efficient activation signal. Here we're plotting the inner-outer average intensity ratio as a measure of activation, and you can see that we also did an IL-6 and an interferon gamma concentration response curve, as well as a time course of induction. And we found that the IL-6 activation of phosphotyrosine STAT3 was relatively transient, and so we picked 20 minutes as the optimal time. 15 to 20 minutes is the optimal time of induction for STAT3, and we used 30 minutes for the activation of STAT1 with interferon gamma. We also did DMSO tolerance, and we decided based on these data for both assays that we would keep the DMSO less than 0.5%. And I think, in fact, when we ran the screen, we were running at a compound concentration of 20 micromolar, which would corresponded to about 0.25% DMSO. STAT3 is phosphorylated, as I said, on the tyrosines, bioreceptor tyrosine kinases, JAKs, SOC, and the STAT family tyrosine kinases. But there is also serine phosphorylation, and there were antibodies that work with total STAT3 that were independent of its phosphorylation status. So we had to look at all three antibodies to see which one gave us the best at the signal window, and we could use for screening. Our bias was to use the tyrosine phosphorylation because it was the main driver of dimerization, translocation, and DNA binding and activation of the STAT3 response. And here I'm just showing you the color composite images of the three antibodies: total STAT3 antibody, DMSO treated, and IL-6 treated, the tyrosine 705, and then the serine 727 antibody. And all three antibodies work well for us. However, when you look at the quantitative data, and here we're using the average inner intensity plotted on the left-hand Y scale, and then the cell count plotted on the right-hand side of the scale, Y-axis. And we're plotting both in blue the cell counts and the total STAT3 average intensity in the nucleus. And we can see that the IL-6 in all three cases induces a nice nuclear translocation, but the assay signal window is much more robust and larger using the phosphotyrosine 705. And since that is the major activating signal for STAT3 for that pathway, we decided to go forward with that as the high content screen. We tested a couple of well-known Janus kinase inhibitors to show that our assay would pick up, and we did We tested pyridone-6 and AZ1480. They're both Janus kinase inhibitors. And we did both a 1-hour compound exposure and a 24-hour compound exposure. And based on this data, we thought that 24 hours, at least, it didn't make much difference for the AZ compound, but at least for the pyridone-6, it looked like a inhibition was more robust and it looked more sigmoidal with the 24-hour compound exposure. And then we tested both of the inhibitors in both the STAT3 activation assay and the interferon gamma STAT1 activation assay. And this is one of the issues with the Janus kinase inhibitors, is that there is a lack of selectivity. So although they're both, they were very potent, about 30 to 40 nanomolar inhibitors of IL-6-induced STAT3 activation, they're also 20 to 30 nanomolar inhibitors of interferon gamma-induced STAT1 activation. And so they would not meet our criteria of being a selective STAT3 pathway inhibitor. So this is just a quick summary of the MCS protocol. Harvest cells, C2000 and Cal33 head and neck cell tumor cells in the assay plate. On day 2, we exchanged the media for serum-free media. On day 3, we pre-exposed the cells 20 micromolar compounds for 3 hours. Treat the controls and the compounds well for IL-6 for 15 minutes. Then we fixed and stained permeabilized cells and methanol ad blocking buffers. Next day we would stain the cells with primary and secondary antibodies, acquire the images on the IXU, and analyze them using the translocation enhanced image analysis module, and then do QC review, process, and analyze the data. This is just a cartoon from one of our publications that really shows we have instigated an assay validation, a couple of tests. One is the 3-day assay signal window test and the 3-day 5-plate DMSO validation test. And I won't go through all the details here, but we use that data and the statistical indices that you generate from that data to help select the data normalization and active criterion for the screen. And then, of course, we incorporate a multi-level statistical and graphical quality control review to QC, pass, or fail our plates. This is just the data from the 3-day assay signal window, Z-factor determinations, doing 2 full plates of our maximum control, 3D full-well plates, and 3 independent experiments. And you can see we've got good, greater than 5 or 6-fold signal-to-background ratios, and on most days the Z-factor was above 0.5, although on day 3 there were a couple of Z-factors that were below 0.4, but we were pretty happy with the data. This is cell-based high-content assay with a lot of steps in it, so we proceeded on. This is how we set up our plates for the screen. We had 32 minimum controls that received 0.2% DMSO and 32 maximum controls that received IL-6 and 0.2% DMSO. And of course, all of the compound wells also received the IL-6. And so this is just a heat map of the plate. This is the data from our 5-plate DMSO validation experiments. And you can see, and we've got the Z prime factor and the signal-to-background ratio and the false positive rate calculated for running the 15 384-well plates. And also you can see a scatter plot, which is an overlay of 5 of the 384, one of the independent runs. And so obviously the false positive rate wells were wells that probably either did not receive either the primary or the secondary antibody. And we could see we had a false positive rate of about 0.23%. We're pretty happy with the data, though, and the robustness and reproducibility of the assay. This is just showing you that if you take the DMSO data and plot it, it very closely approximates a normalized distribution. And we did not see any row or column effects or positional biases, so we felt pretty happy with this and proceeded to go on to the screen. And to finish the validation, we tested the low-pack set, 1,280 compounds, and the 446 compound clinical collection. Screened them at 20 micromolar, and we set our active criterion as a mean percent inhibition greater than 50%. And we were calculating that based on the IL-6 and DMSO plate controls, normalizing the average inner intensity of the phosphotyrosine 705 signal in the nucleus. We cherry-picked the active compounds, and then we retested them in triplet wells at 20 micromolar against both IL-6-induced and interferon gamma-induced activation. We did a 24-hour compound exposure prior to cytokine activation. And this is just a summary of the Z-prime factors and signal background ratios. We ran the assays twice, and everything looked pretty good, although we were pretty surprised to find such a high hit rate. And so this is some reason for concern. So then we went and looked, we used our outlier analysis, and we looked at the cell counts, and we identified compounds that had less than 200 cells per image, and we classified those as cytotoxic. And then we looked at the Hertz nuclear intensity Z-score, as well as a Z-score for the channel 2 total and average outer intensity and the total and average inner intensity. So we looked at all the fluorescent outputs and tried to identify fluorescent outliers. Even so, we ended up with a pretty high hit rate, and this is an Excel table showing you the PSTAT3% inhibition and the PSTAT inhibition that we detected. And you can clearly see, if you break down and try to color code the agents in the low-pack set, that there were 23 beta-adrenergic receptor activators, as well as the dopamine and H1. And what's interesting about these is they're all GS-coupled GPCRs. And but it was troubling that we were getting this many. But then we went to the literature, and we had a 2.9% hit rate. But then we found out that there's a full body of literature saying that suppressors of cytokine signaling family, SOX1 to 7 and CIS, that are inhibitors of cytokine signaling, they're classical feedback inhibitors, and they block STAT3 signaling by binding to and directly inhibiting the Janus kinases, by competing in the STAT3 phosphotyrosine binding sites on activated receptor chain, and also targeting the signaling proteins for proteasomal degradation. There's a fair body of literature out there that say that agents that elevate intracellular cyclic AMPs or activate protein kinase A will activate SOX3 expression levels independently of the STAT3 pathway. So any GS-coupled GPCRs that elevate, or agents that elevate or activate PKA will upregulate SOX3 levels and inhibit STAT3 activation. We've actually since gone on taking one of the beta-adrenergic receptors in horse colon and been able to show that indeed we do see upregulation of SOX3 with those agents. When we took the hits and we did a comparison between the confirming the inhibition in triplicate wells of the inhibition of STAT3 and comparing it to the inhibition of STAT1, we found there were only 5 of the hits were actually selective for inhibition of STAT3. I'm showing you the structures here, as well as if you do a PubChem cross-target query. This data is a little bit out of date, but if you went in and used the PubChem SIDs, you can find how many assays they've been screened against or how many active flags there are in the database. So we took each of those compounds in and we did full concentration response curves, and we did a number of time points of exposure. We did 1-hour compound exposure, 3-hour compound exposure, and 24-hour compound exposure. And I'm just showing you the relative data here. I'm plotting the percent inhibition of IL-6-induced STAT3 activation on the left-hand x-axis and cell number on the right-hand y-axis of each of these graphs. And you can see that at 1 and 3 hours of compound exposure, you see good inhibition with minimal effects on cell numbers, whereas at 24 hours exposure, you're seeing a significant inhibition or reduction in cell numbers. So based on these data and the earlier data with the Periodome 6, we changed our screening strategy, and we decided that we would go with a 3-hour compound exposure rather than either the 1-hour or the 24-hour that we had done for the low-pack screen. And we thought that this was a good compromise. So we identified more inhibitors, but not have to deal with a higher level of cytotoxic compounds. Here's just the summary data showing you that we went back in and tested some of the low-pack hits and full concentration response curves. And you can see that the itraconazole and the etomidate did not repeat in concentration response curves. But the isoepirine did repeat, but it was not selective. In fact, it was very, very potent and consistent with it being a beta-adrenergic receptor agonist. There was no selectivity for inhibition of STAT3 or STAT1. Apolilactin was the only hit from the low-pack screen that was confirmed. It has a relatively modest IC50, 26 micromolar, and greater than 50 micromolar. In addition, we took the 4 head and neck cell lines that we had before, and we ran growth inhibition assays using cell cycle flow detection reagent, and we got pretty reasonable GI50 data with apolilactin. So this is how we ran the screen. We screened in 56 wells at 20 micromolar, 3-hour compound exposure, IL-6. We set our active criterion as greater than 70%, and then we used the multi-parameter HGF data for outlier analysis. Here's a summary of the screen. We had 1,000 hits, and then we eliminated, you can see the numbers that we eliminated for cytotoxicity, channel 1 and channel 2 outliers. The average Z-prime factor was 0.47. Over the almost 384 well plates in the average, signal to background ratio was about 4.7, so the assay performed well on the screen. This is just a summary. We did concentration response curves against both PSTAT3 and PSTAT1. We had 203 compounds that produced an IC50 less than 50 micromolar against STAT3. 88 compounds also produced an IC50 against STAT1. And we had 54 compounds that produced IC50s for STAT3 activation less than 25 micromolar, and that were greater than or equal to two-fold selective. So the chemists took a look at those 54 hits and they selected chemically tractable hit compounds. And we looked at those compounds and did a hit characterization. These are just some images using the total STAT3 antibody. You've seen data like this before. We use the multi-wavelength self-scoring image analysis module to quantify the total STAT3 intensity in the cells, both in the nucleus and in the cytoplasm, as well as the total cell. Here are the data outputs. You get cell counts, and then you get mean nuclear intensity, mean cytoplasmic intensity, mean total cell intensity, mean integrated nuclear intensity, integrated cytoplasm intensity, and integrated cell intensity. So IL-6 in treatment induces increased levels of total STAT3 in the nucleus and a decrease in the cytoplasm. So it can significantly affect the integrated total STAT3 in the whole cell. And we took the 10 hits from our screen, and we looked for effects on the total amount of STAT3, because it could be depressing the amount of STAT3 or causing degradation of STAT3, total STAT3 levels in the cell. And as far as we could detect from these data, that none of the 10 selected hits significantly affect STAT3 protein expression levels. The chemists selected 4 hits from this series. We did some additional, Jenny did some additional cell-based assays, and those compounds are now going through a lead optimization, chemistry lead optimization, headed up by Donna Thurin's group here at the University of Pittsburgh, the UPCDC group. This is just showing you our workflow, image acquisition, analysis, and then loading it into the store. Pull it out into Spotfire for data visualization, and everything is tracked through activity-based. This is our setup. As I told you, we have the PowerCore client software to do the distributed computational analysis. Really speeds up the analysis for us. And we have to have the gigabyte Ethernet type because we're transferring a lot of data. We've got the store, the MDC file server, and the PowerCore client. You have a secure network architecture because you have to obviously have enough space to store all the data. And just to illustrate that, 1 384 well plate, the way we screen it, has 2.88 gigabytes of space. A single run of 19 plates has 54.9 gigabytes of space. When all said and done, when we finish the screen, we have 1.66 terabytes of space used on our server to accommodate the images and the metadata for the here are the acknowledgments. Jenny Grandis is the PI of the project. She was the next applicant that got it funded. John Lago was co-PI of the Pittsburgh Specialty Applications Center, which is part of the NCI's Chemical Biology Consortium. Yun Rod, Dan Comerico, Tong-Yin Shin, and Harold Patsy were part of the project team. The chemistry team that generated compounds for us and are currently heading up the lead optimization are Donna Thurin, Peter Witt, and Lynn Resnick. And they have additional chemists that have been added to the team for the lead optimization. And from the National Cancer Institute and SAIC, we have Shizuka Say, Sanjay Malhotra, Beverly Teicher, Joe Morris, Joe Marcusson, and Barbara Wachowski. The funding for this is listed here below. The NCI funded this as a contract. The numbers listed there. The next PDCI project ID is number 105, and this was Jenny Grandis was the PI. And I'd be happy to take any questions.
Really, really great. We'll let give people a couple minutes to write down their questions. Actually, I'll start while people are thinking about it. Why did you choose the an HCS rather than some other technology like gene reporter or reporter assay to run this screen?
Well, it was a novel approach, and it gave us the ability to run the STAT3 or the STAT1 high-content screen as a counter screen. A lot of people had used reporter assays and fluorescence polarization assays to screen for STAT3 inhibitors. And to our knowledge, none of those have really evolved into compounds that were moving forward into clinical application. So we thought we wanted to take a novel approach, and really because we had defined that we wanted to be able to do the assay in a relevant tumor cell model, and to Selective the head and neck cell tumor cell model, and to be able to run the counter screen for STAT1 with all of it.
Somebody asked a different question. Were STAT3 and STAT1 assays at the same time on the same plate separately, or did you also did you do them all in parallel?
We actually developed methods to do them all in parallel, but when we got right down to it, there were issues validating both assays, doing them in parallel. So we actually ended up running them in the same cells, off the same compound plate, but actually fixing and staining them independently. So we did not run them in parallel. Well, we were able to develop methods where we could run both STAT3 and STAT1. And actually, when we ended up, the reason we didn't run them in parallel also was that the species for STAT3 and STAT1 antibodies, the fast antibodies, they were both monoclonal, so we couldn't do them in parallel in the same way. However, we were able to identify combinations of other antibodies, but they weren't quite as good as the monoclonals. So that's why we actually ran them independently. Our original idea was to run them both at the same time, but we ended up not doing that.
Okay, the person asked a follow-up question. When developing the assay, did you have difficulty with generating a large enough delta between high endogenous nuclear T phospho-STAT3 in cells with consistently active activation of STAT3 and IL-6 induced signal in the same cells? Did you understand the question?
Yes, I did. It's a very astute and good question. Actually, that is why we serum starve the cells for 24 hours to suppress the endogenous T-STAT3 levels, but still maintain a robust IL-6-induced STAT3 activation.
So yeah, I actually have my own clarification on doing the counter screen. Did every compound go through your counter screen, or did you only take your hits and run those through the counter screen?
We took only the confirmed hits. So when we did concentration response curves, so what we did, we actually ran the screen, identified the hits, we cherry-picked the hits. We did confirmation at the 20 micromolar screening concentration, and at that stage we ran both STAT3 and STAT1, and then anything that confirmed, we did then 10-point concentration response curves in triplicate.
Somebody asked, was there any advantage of running this assay with the control group?
We actually were able to validate and run the assay on a wide field imaging platform, the ArrayScan BTI. So, no, I really don't think that the confocal was required. But that being said, it ran very well on the confocal platform.
Okay. Any other questions? Are there any other questions from the audience? Okay. Just give it a minute. Paul, again, I really appreciate this. That was great. Yeah, somebody that says thanks. Yep, anybody else online? Somebody asked, yes, I should have mentioned, somebody asked will the recording of this webinar be available? Yes. Within about a week, the WebEx will edit out the beginning and the end and make sure there's no mistakes at the beginning and the end, and we'll post within about a week to 2 weeks. And then we'll send an email out to both the people who attended as well as those who didn't attend, letting them know that it's posted. And it's posted on the the moldev.webex site, which there'll be a link when we have that. And there are other webinars available, which we'll also let you know about. Well, thanks everybody. All I appreciate it. We'll be touching base later. Thank you.
Okay.
If anybody has any questions, just let me know. Email me.
And if anybody wants to email me a question, they can also do that too.
Okay. And if you don't have Paul's contact information, you can get it through me.